And big hand of applause for Dr. Kim. Thank you.
Okay, so it's very difficult coming after Dr. Lee, Justine. So as she's dynamic, and I'm only going to, I only know human and skin. So I hope what I'm going to be saying to you is exciting for you, at least interesting. So let's go on our first slide. Okay, so first of all, thank you very much, especially to Jolie for inviting me here, and thank all of you for actually staying the very last talk. And my talk is very different than what you just heard. She told you stories, what she went through. Mine's a little dry. I thought he was going to say the slides freaked him out because of the basic research, but I think he was talking about the photos of dermatology. So thank you for being here. Thank you also because in my household, my status just went way up. I have 2 teen daughters. They don't quite understand why I practice human medicine, but they do, especially my younger one. If there's anything that she would do in the world is to be a vet and a rock star. So Purple Rain, my gosh, I should have brought her here. So totally LA girl. And she's asked for many, many dogs. And no, we only have 1 or 2 at a time. Then she started asking for horses and then cats, where my older girls is allergic, then asked for bigger cats as if it meant that she wouldn't be allergic to it. Then we moved on to pigs because there was a man in Brentwood walking a pet pig one day. And then I said no, and now porcupine. But in California, I believe porcupines are illegal to keep as pets. And then we moved on, and now she would like to meet large rabbit. And these giant rabbits apparently live in Malibu. So this summer, we are going to make a trip to meet these giant, large rabbit. So that's sort of my household. I wanted to talk to you about skin and human skin, really, that's all. But then when I started to say, what can I absolutely tell them? It's going to, I know deep in my heart, skin is a beautiful, beautiful organ, and it's amazing. But I said, okay, if human skin's that amazing, what about animals? And I don't know anything about animal skin. And then I started seeing there's some amazing animals with amazing skin. Like, did you know that crocodile have sensitive skin? I didn't until a few weeks ago when I started sort of searching. Sperm whales have thick skin, and what my patients would do to have thick skin, they wouldn't need to put fillers in their face. African spiny mice can regenerate very well. Now, I may be saying all this wrong, and tell me if I'm wrong, but thorny devil, apparently they have skin that will absorb water or fluid like crazy, and so they're known as living paper towel. I didn't know that. And then the giraffes with air conditioner skin. So when you're nervous like me, maybe you'll cool off. And cephalopod have eyes on their skin. So animals also have amazing skin, but I'm going to be focusing on human skin. Now, for those people who are not in dermatology or skin research, we at UCLA, the cardiologist, the neurosurgeon, they sort of poo-poo us. Skin, just the wrap, but it's really not just like a saran wrap. Skin has amazing, amazing potential. It is one of the largest and important immune organ of our body, and has complicated structure and function. It's really a fingerprint of what's going on inside that sometimes manifests to the outside. So skin can tell a story about a person. So I'm going to outline structure of the skin, of human skin, how it functions as a barrier, physiology a little bit. Sorry for the boring talk, basics, and the amazing mechanism that our human skin has, and focusing on immunology because that's my area of expertise, and then talk about a few very common skin diseases that's related to immune system. So as you can see here, skin human skin is about 100 micron, and they're layers, amazingly made, with the basal membrane that separates the epidermis from the dermis. And even the epidermis has very different types of cells. They're all keratinocytes, but the first layer is called basal layer, and then they differentiate, and then they start to produce keratin that are very different. As they become more differentiated, they start to form keratin hyaline granules, and I don't know if animals also have that, but those are the granules. And later, as the skin matures, even the dead skin that loses its nuclei and becomes stratum corneum, they have a function because they start to produce certain lipids that will give our body protection. So pretty amazing, I think. The most common skin or cell within the epidermis are keratinocytes, as you can see here. And people used to think that keratinocytes were just sort of cells that just sat there. And that's not true. We know that they produce keratin, but they also express many, many molecules and receptors that we can respond to. And that makes sense because our skin is the first thing that's exposed to the external environment. So they do respond to microbe and other things to mount an immune system, which I'll talk about a little bit later. Another common cells within the epidermis are Langerhans cells, and these are really actually amazing cells. They have little dendrites, and they have special receptors and even have class 2 antigens receptors. So what they can do is they can actually recognize antigen, take it up, process it, and activate your T cells. So here it is. You have an antigen on the skin surface. These dendrites or dendritic cells called Langerhans cells recognize the antigen. They take it up, move down to the draining lymph node where they'll present what they saw on our skin to say, come on, activate. So T cells can go back and fight infection. Pretty amazing if you think about it. Third cells that we see in the epidermis, melanocytes. So melanocytes are there's one melanocytes per about 36 keratinocytes within human epidermis, and they give us the color of our skin. Now, whether you're light or medium or whatever color you are, basically in humans, everyone has about the same number of melanocytes. What's different in color giving are the melanosomes and type of melanosomes. So if you're a little bit darker, you might have eumelanin, brown or black melanin, and they absorb and protect your skin a little bit better from UV. If you're fair, like blonde or red hair, then you have more of a pigment called pheomelanin, which they don't function as well in terms of protection against UV. So physically, skin functions as a barrier. So you can impede things just physically. They absorb and block solar and ionized radiation. They inhibit water loss, and they're able to control the temperature. And even the stratum corneum, where it's sort of a dead skin that turns over, they have lipids and oil and things like that. Certain epithelial areas in our body, like the gut, or the airway, we have special functions such as having a different type of enzymes or acid secretion, or even physically might have a ciliated epithelium where we can utilize these structures to protect our body. So really pretty amazing. Now, when things go wrong and when that turnover from the basal layer to the stratum corneum doesn't function well, the average time your turnover is about 28 days, then disease starts to occur for humans. So this is a patient with epidermolytic hyperkeratosis. And these patients, they can't turn over their cells within the epidermis. So you can see how thick the stratum corneum is. It's just as thick as epidermis. And so they start to have thick skin, crusty like that, and they get recurrent bacterial infection, these humans. The other side of it is patients who don't or can't have skin for whatever reason. Is a patient with TE and toxidipperlysis necrolysis, And this occurs maybe due to infection. Another common problem of this is sometimes medications of some sort, sulfa. And we get called, very few times we get called in to emergency room as a dermatologist or in the hospital. But TEN, Stevens-Johnson syndrome, is when we do. And if you don't have skin barrier, then you get dehydration, infection, sepsis, and patients can die. High rate of mortality with this syndrome. If you don't have melanocytes, then you can have pigment loss, like this young man with vitiligo, and that leads to chronic problem without being able to protect his skin against UV. So exposure for vitiligo patients can lead to skin cancer development and other problems. Now, this is an area that's really amazing. Recently, it's been shown that, clock genes are present. And whoever thought, we hear about clock genes, when you go on a plane and you have time change, we take melatonin. Well, it turns out that clock genes are also present on skin. So, and that makes sense, right? The skin is out there, first clue to interacting with the environment. So you get an input, goes to the central clock in our brain, and then there are peripheral clocks. And it turns out in the skin, some of the physiological functions, such as water loss, temperature, and sebum production, is regulated diurnally by clock genes. So this was an article in 2009, New York Times called, and they wanted to do an article on clock genes. And I used to wonder, those cosmeceutical companies, why does anyone need a night cream? Just put the day cream on at night and night cream at the day. Use it twice. They do have some, and I used to sort of poo-poo it, but they do have some research that kind of justifies why there's a night cream. So it turns out that physiologically, you have different water loss at different times throughout the day. You also have different temperature regulation at different times of the day. So you have highest temperature sort of at midnight, and then sebum production is highest at noon, but lowest at night. So if you can regulate, and there are therapies now for cancer, if you can utilize this physiological function of your body and skin, especially if therapy is through your skin, then one might be able to treat or improve skin health better in this way. In my laboratory, we found that there's a gene, actually a clock gene called BMAL1. So if here's a BMAL1 making a lot of, these are just sebocytes, making a lot of lipid drop, as you can see here. If you knock that gene out, the sebocytes don't make any more lipids. So it's really interesting that we can regulate the physiological function of our body through clock genes. Another thing that skin can do very well is make vitamin D. So it's an amazing organ that makes our vitamin D. When you are out there enjoying the day today, during the break, we all got a little bit of vitamin D. So skin is a place where sun hits our skin. We make vitamin D3, and then that goes through our body, through the liver and the kidney to make active vitamin D. And it turns out that no matter how much food you take, the best way, the most efficient way of getting your vitamin D is sun. Now, American Academy of Dermatology and myself, we don't promote that you go out and tan or get sun exposure, but knowing this is important because it turns out that cod oil is probably the highest, but who wants, in vitamin D, who wants to take cod oil? In salmon, especially the wild salmon, has a high level of vitamin D. If we always tell our kids to drink milk, and it does, but it's not really that high. And 90% of vitamin D that we can get come from sun and very little from our food. So you do have to take supplements. Now, FDA and U.S. government says, you need about anywhere from 200 to 600 IU per day if you're an adult. But it turns out that through a lot of vitamin D research that we and other labs have done, for immune function, you need much, much higher vitamin D level. So if we need around 2,000 to 4,000 IU per day, that means you have to drink about 20 to 40 glasses of milk, which we won't do. So taking supplement is one way that we can get our vitamin D. besides these physiological functions that I talked about, we have incredibly sophisticated mechanism to protect our body. So first thing for our skin, the most damage that we get is UV. So on Earth, because of the ozone, we don't really get UVC exposure. But UVA and UVB are what our skin can get damaged from. UVB causes immediate sunburn. So if you go out in the sun, you spend the day at the beach, the redness that you get is mostly from UVB, and chronic sun exposure is from UVA. So this is a truck driver. You can see his left side of the face. Years of sun exposure on the left side leads to that, and the right cheek looks much younger. Both UVA and UVB are responsible for development of skin cancers. So I told you earlier, the stratum corneum can reflect the UV, melanin can absorb UV, and then sequester the reactive oxygen species. And then when cells are damaged, we have an incredible mechanism to kill the bad cell off, and that's through apoptosis, as you know, so the sunburn cells. But in the skin knows when the damage was done by sun. So when your skin cells are hit by UV, the DNA is damaged and they form a very specific photodimers. So the body knows, oh, this pyrimidine dimers or the 6,4 photo products are due to sun. And then that damaged DNA can be repaired by our DNA repair mechanism. So here's that little red spot where the sun damaged the DNA. So the enzymes that we have in our body comes in, it excises that damaged area, and then we transcribe better DNA. So this is happening every single time you're going out there. And obviously when that goes wrong, then we start to develop precancers and cancers. Now, not everyone has normal mechanism. So this is a young girl with xeroderma pigmentosum, and this girl does not have an enzyme that can repair the DNA damage. So then she starts to develop skin cancer at a very, very young age, and every single time she goes out in the sun, she's damaged. So she has to go to school during the night, or her school, actually, they were very kind to put a dark shield all over the window, and then when she goes out and plays, it's really at nighttime. Otherwise, the sun will damage her because she doesn't have the mechanism that we all have, that we take for granted, really. So I learned that, and tell me if I'm wrong or not, I would love to talk to you about it. Apparently hippopotamus skins are amazing in terms of antimicrobial response and sun protection. So they have two sort of pigmented molecules, one called hipposudonic and the other orange molecule called norhipposudoric acid, and they're supposed to be protective, and these molecules are supposed to induce antimicrobial response and UV protection. So I don't know if these are being developed for pet use or human use, but how interesting, because some of the most amazing treatments that we can see or we can develop already exist in our nature. This is another mechanism. We talked about UV excision and repair. Now, every single time we hit our skin or something happens, we injure ourselves, we're not always rushing off to the ER and seeing Dr. Lee, So that's because we have an amazing repair mechanism. So you get your cut, and most of the times it heals beautifully. The wound healing process is really complicated, but very well thought out, whoever thought of this, and it's an amazing mechanism. So initially, within minutes, hours, you start to get inflammatory cells to the local area, and then the proliferation phase starts after days, where the keratinocytes try to come together, and then after that, from weeks to months, you get sort of the remodeling phase of collagen and so on. So most of the times when you have a cut, then it will repair itself. The second column is a patient where she had Mohs surgery for skin cancer, and we need to do some bilobe flap to fix it, and she turned out pretty well. But we did all this fancy work, and another patient with also basal cell carcinoma, we left it alone, and it healed almost as beautifully. So really, our skin is an amazing way that it has inherent ways of healing. It's not true for everyone. So patients with diabetic, they can't heal very well. Their wounds don't heal. This is a patient with diabetic ulcer, especially the lower legs. So it turns out up to 43 patients of diabetic ulcers don't heal, leads to many, many complications and mortality, and can get amputation if this doesn't heal or leads to bone infection. I'm sorry to show you this. I know it's really, really hard, and I don't know if animals get epidermolysis bullosa, horrible disease where collagen 7 is missing, so epidermis and the dermis don't attach together. So it's like having chronic erosion on their body every day. Because of the sort of embryonic development. You know as well as I do that the skin comes from the ectoderm involving the esophagus and the GI tract, so they can't eat at times, and we have to put a G-tube in. And these are, I take a lot of these, care of these patients, not through my practice, but through a foundation I found. And we have a camp for these kids and other kids with very, very severe skin condition. So these are some of the campers at our camp, and most of them we have to wrap them up, and it takes us 4 to 5 hours sometimes to wrap each kid every day. And this is how they live until sometimes they pass away in early 20s or so. And so if we can learn from these little spiny, African spiny mice, how they regenerate, perhaps we can develop ways to improve wound care, not just for little cuts that I or you might get, but for some of these kids, diabetic patients, or kids with epidermolysis bullosa. So I think we can learn a lot, and that's what I'm hoping that we can share knowledge with each other from. So another mechanism that our skin has is our immune system, and it is beautiful. Our skin is the best immune organ of our body. It's kind of a complicated slide, but basically here's a cartoon drawing of the epidermis. The keratinocytes are really immune cells. They're innate cells, so when they see the foreign object that they want to get rid of, they can mount an immune system. Non-specific immune system to kill the bacteria or bug or induce inflammation. So our immune system, as you know, can be divided into 2 separate immune systems. The innate and the adaptive. And innate is something that's innate to our body. You hit the bacteria and you're going to kill it. You get injured and you're going to repair. And that requires receptors to recognize those pathogens and then induce inflammatory response or antimicrobial response. Then the innate immunities and immune cells instruct the adaptive immune cells to get better at what they might be more specifically doing. So T cells and B cells are adaptive immune response, and they give us memory. So if you have an infection or a cold or something bad that's happening, it's really our vaccine, right? They remember so that if you get exposed to something really dangerous, then you know how to take care of it. So those are interaction of innate and adaptive immune response. Ooh, I think they want us to stop. I think we lost our slides. There we go. So this man, Elie Metchnikoff, won the Nobel Prize in 1905, I believe, for physiology. And he's the one who detected that our innate cells called phagocytes can mount an immune system, innate immune system. And he used the starfish larvae to study this. So we know that now, not just the starfish, but this fly, Drosophila, also have these amazing receptors. This Drosophila can tell through 2 receptors what's fungus and what's bacteria, and that's all he needs to know. And when he recognizes that, he has an immune response that can kill those things off. And humans are no different. So these are innate cells in our body. And what humans can do is that we also have these receptors called toll-like receptors, just like the toll receptors in Drosophila. So your body can tell, is it a bacteria, is it a virus, and it's even a little more specific than that. Is it a DNA or an RNA from bacteria or viruses? So it can recognize it and then activate our innate immune system to quickly defend our body. And this is all present in our skin. So when the innate immune system recognizes it and quickly responds, secretes cytokines, it can mount fever or healing process, or it can induce cytokines to instruct adaptive immune cells to come on, get to work, B cells and T cells, and that's what they do. B cells then produce immunoglobulins, and T cells then decides which way do I want to become. And T cells can become T helper cells or T cytotoxic cells. And these T cytotoxic cells are what kills your tumors, or T cytotoxic cells are what kills bacteria-eaten cells. The helper cells are, again, the instructors of your immune system, and it will tell us how to take care of our body. So here's another cartoon drawing. Let's say in the skin you saw a bacteria that Langerhans cells brings it to the lymph node, and then the T cell starts to decide which way do I want to be, what kind of T cells do I want to be, and then go back to the skin. We now also know that there are things called T regulatory cells. So just look at your skin, or look at your partner's skin next to you. It's pretty amazing. I mean, I study inflammation, but to me the opposite is more interesting. Why is your skin so beautiful? It should be inflamed all the time because we're exposed to something. So we have some kind of immune regulatory cells, and we're finding out now, this is very recent work, that there are T cells called regulatory T cells that will keep you nice, skin, beautiful all the time, so that just because there's toxins or whatever it's in the air, we're not just reacting to it immediately. So what happens when these T cells and immune regulatory goes wrong? I just talked for a few minutes about how amazing skin immunity is, but things go wrong. And when things go wrong is when disease occurs through immune system. A very good example is something called leprosy. I don't know if animals get leprosy, but humans do. In fact, in Los Angeles, we have a big Hansen's disease clinic here where we see leprosy patients. Now, M. leprae, Mycobacterium leprae, is the same bug, right? Mycobacterium. For some people, they have lots of TH response, so they only get small skin lesions. For other people, it's the same Mycobacterium, but they mount a different type of immune response, and they get disease that's very, very severe. Their nerves degenerate, and they start to form plaques everywhere. So how come we're responding differently? Why do some people respond differently and others don't against the same Mycobacterium? And that's what we want to study. Same thing with disease, and I don't know if that's the way in animals, but certainly in humans, psoriasis, skin cancer, acne, it's all dysregulation of immune system. This is a patient with psoriasis. Approximately 2.6% of our population gets psoriasis, and it's a thickening of disease where we now know that it's not just cellular proliferation, but also alteration in the immune system. So there's something wrong in psoriatic patients with their Langerhans cells, those dendritic cells I told you about earlier, and there's something wrong with their T cells. They have too much Th1 and Th17 cells. So people used to say these are infectious patients, don't go near them, how do we treat them? We now know that it's immune response, and we're trying to treat these patients now with immune therapy. So when the antigen, whatever antigen is, we don't know what psoriasis antigen is, but same pattern here, something in the skin goes to the lymph node, activates T cells. In the past, I told you they activate because they want to protect our body, but now they're being activated to some unknown antigen, and then the T cells, especially the Th1 and Th17 cells, go crazy, and they're just all over our body. They come back to our skin and cause disease. So now therapy is different. And then they also see cytokines in the local skin. So the therapy really, let's see, is using immune therapy. So we're blocking T cell response so that these patients cannot mount excessive Th1 or Th17 response. So anti-TNF alpha, anti-IL-17, these immunotherapies are what we're using for patients, and we're seeing pretty good response. Whereas in the past, we were using methotrexate, cyclosporin, and a lot of things that were really harmful to our body. The second immune-related disease in human is skin cancer. So skin cancer, melanoma is the most serious, about 140,000 cases of melanoma. And it turns out that in humans, it's the second most common disease, cancer for a young woman between age 15 and 29. So very serious, and death rate is very high for melanoma patients. This is very different than non-melanoma skin cancer, which is usually due to sun, and there are about 2 million cases per year that we see, mostly being basal cells. Here, for non-melanoma skin cancer, mortality is very low, but morbidity is high, because I showed you earlier the Mohs surgery that we did for skin cancer. You know, we do this, thousands of these each year at UCLA. So same thing, our cytotoxic T cells should be fighting skin cancer and trying to kill those things off. But at some point, when the immune system can't control it, then your skin cancer and other cancers, for a matter of fact, just can spread. So one way that we're thinking about is, you know, I told you these regulatory cells that keeps our skin really nice. When that works too well, it's working when it's not supposed to, like when cancer's there. So it's this sort of balance of how do we up the good balance and how do we down the bad balance. But the bad and the good depends on what each situation, right? When it's infection, you want one to be high and one to be low. But in cancer, it may be the other way around. So regulating these T cells' immune response is important for cancer treatment right now. In addition, cancers like little virus, they mutate, and they can just change to get away from your immune system. So for example, this is squamous cell carcinoma secreting interleukin-10, which is inhibitory cytokine. So it's trying to get away from your immune response. So we're using immunotherapy now for skin cancer. Imiquimod is one thing that we're using. I don't know if it's used in veterinary medicine, but it activates dendritic cells to secrete interferon, and that's how we're treating skin cancer, so we can avoid some surgery. Melanoma is really difficult to treat. Just recently, we made some headway. It turns out that up to now, for the past 30 years, we cannot really improve the survival of anyone with melanoma that's metastatic. Just recently, with a combination of multiple immune therapy, for first time we're showing that we could improve their survival. So this is really, really huge advancement, and it's with these two treatments. So one way is to block one response, CTLA-4, another way is to block PD-1. Both of those receptors were telling our immune system not to work. So by using antibody against those, now we're getting rid of inhibitory response that exists in our body, and it's regulating our immune response. And now it's showing pretty good response, and this is going to become more of a standard of care for really metastatic melanoma patients. And so this is really exciting time for treatment of skin cancers using multiple immune therapy. Now, the very last disease I want to talk to you about is acne. And this is something I hope I'll get to talk to you, some of you, after my talk. I don't know of any animals that get acne, and it would be really interesting to know if you know. Cats? Oh, so maybe we need to collaborate with cat doctors. So it turns out in humans, as you know, almost 80 to 90% of people get acne. It's so common that it's almost like, is it really a disease? And while it's not, there's no mortality, and people not in dermatology think it's not a big deal. It's a huge big deal. First, it affects so many people at a very important age in their life. Kids that I've met, they quit school. They don't want to socialize, and it brings their self-esteem down, so much so that it affects them into their adulthood. So we're trying to understand this disease because it's really not infection. And despite what people used to tell us, it's not because we're dirty or because we eat chocolate. And it turns out that it's really these factors. There's something going wrong with our keratinocytes proliferating, also oil production, sebum production, but mainly P. acnes and inflammation. So now it's thought that acne is an immune disease. This one slide is 10 years of my life in the laboratory. Yes. So what we found in our lab is that P. acnes, the bacteria that's associated with acne, binds to a receptor called toll-like 2 receptor. Our body recognizes it and induces cytokines, and then it breaks down your collagen. So that's why when people have acne, they get scarring. Not every severe infection of our body leads to skin scarring, but acne does. So this is one way, this is a mechanistic pathway that we worked on. And so we're trying to, again now, we never used to think of treating acne with immune therapy, but it is an immune disease. And so this is a new way of thinking that we're trying to see, should we not use antibiotics? Should we target immune response? Should we block those immune responses? Also interesting in our lab that we found is that not all bacteria are the same. So the P. acnes that we find maybe on your skin versus your skin or my skin are all different. We can phylotype them and ribotype them, and it turns out that some are associated with acne, even though they're same bacteria, and some are not. And what that means is that these P. acnes induce different immune response. So the red marks are P. acnes that's associated with acne. They produce lots and lots of interferon response and IL-17 response, the inflammatory response. The normal associated P. acne induces high level of IL-10, way over there on that end. So they don't mount an immune response. So if they're on our skin, the healthy P. acnes don't induce inflammation, or they induce inhibitory inflammation. So by regulating your skin immune response, we may be able to prevent or treat acne in the future, and that's what we're working on. Finally, I think that you all know very well, probably from Barber Talks and so many of your work, that animals have every emotion and every feeling that humans get. They're like our kids probably, right? So what's interesting is that medically, skin is interesting because it conveys what's going on inside, but also your emotions and other well-being too. So medically, if you have any of these, you can induce certain type of skin response. Patients come in from internal medicine suite, and then they say, I don't know what this patient has, and then we can figure out in our dermatology what they have, because there are some skin lesions that's associated with certain medical disease. Similarly, I think in terms of internal well-being, because beauty is so associated with skin, that it can affect us in so many different ways. And so treating, I think, skin health is very important. These kids have many different type of skin disease: vitiligo, epidermolysis bullosa. These two kids down here have ectodermal dysplasia. And so they come to our camp because they're ostracized by their peers in our society. So it's 1 week where no one thinks that they look so different. But physically, they can't hide their disease like kids with maybe cancer or HIV or whatever else going on inside of their body can hide. So it's really, really, it affects their self-esteem pretty significantly. So I'm grateful that I've been invited to this conference and symposium, because I hope that just like there's so many things going on in terms of comparative health for other disease, I hope that we can start a conversation and collaborate together, human dermatologists and animal dermatologists, because it seems like there could be a lot that we can learn from. I was told that melanoma, for example, is not serious in animals, in white horses. Now, in humans, melanoma, the word is so scary because it's so aggressive. So maybe there could be a gene that we can find that could be different and differently regulated. Same for wound healing and so on. So I hope that there will be more conversation and collaboration going so that we can improve skin health for animals as well as skin health for humans. I want to just end by, it's like a cycle life is, right? I mean, I never thought in my life I would be talking to you in front of you, but I think Dr. Lee mentioned about Demodex. Well, it turns out my first experience with veterinary dermatologists was because of my dog Clivis. He had bad skin, so it was before kids, like he was my life, and I slept in my bed and skin turned terrible, and I was a dermatologist, but I was so scared, like I didn't want to scrape, like she said, you don't want to scrape your own animal. So I took him to a dermatologist and they scraped it and found that it was Demodex. Well, I'm like, we see Demodex, I could have done that, but no, I could not have done that. So they told me to treat him with topical Midoman or something. So, right? So I thought I was being so good. Okay, we want him to get better. Mix it with little cream so that it can be absorbed a little bit more. This was like my first year residency. Put him on, and he went limp. I thought, oh my gosh, what did I do? And I'm like, and then my husband came home, or my husband to-be came home, and we figured out what was going on. He absorbed it too much. So put him in a car, and we drove him down, or I guess I drove him down into the Sepulveda emergency area. And I ran in carrying him saying, he needs yohimbine. And if anybody in human medicine heard, they would have thought, this woman is crazy, right? But they knew exactly what to do and what happened to him. So I think she's right. Sometimes we leave it to you guys to take care of animal dermatology and me for skin dermatology for humans. But what is interesting is the commonality and how much we want the world to have good health for humans as well as for animals. So I really appreciate your time and your kind invitation to have me here today. Thank you.
Thank you very much. Thank you very much. And I'm sure there are some questions. I know, as a matter of fact, before your talk, this is quite interesting, that before your talk, a couple of the veterinary dermatologists came to me after Barb was talking, and they said to me, we need to talk to human dermatologists because there's so many similarities. Mm-hmm. We never talk with them in depth, and so we need to set this up. So there are a couple of these veterinary dermatologists in the room, and I'm sure they're dying to ask you some questions. And I thought dogs did get acne.
Did they?
Oh, look, they're frowning. Okay, can we use a microphone? There is an answer to the question. So I thought, let's answer that question for you right now.
Right.
Anybody have a microphone, please? Oh, there. All right.
Is this on? Yeah. Not in the same way as humans do. And though a lot of people refer to chin conditions in cats as feline acne, even that condition, to the best of my understanding, is not analogous to the human condition. I'm a dermatologist. We've got 3 right here. We've got...
Yes, exactly. Can you introduce yourself?
I'm Brian Scott. I'm a veterinary dermatologist from St. Petersburg, Florida.
Boarded?
Yeah.
Yeah, so...
This
okay,
is
great.
Dr. Nicole Heinrich. She's from Minneapolis. Klaus Loff from Boston.
Okay.
Excellent. Yeah, he gets really red. I can see it. Any other questions? Well, Dr. Kim is here. I have a question for you. So the one talk, I went to another talk, a human dermatologist, and maybe you can explain that to me again, because he was talking about this little spot in between your shoulders that itches, but that is kind of a sign of a different disease. And I thought, oh, that's great. As a matter of fact, what he said was that if you have a neck problem, and so there's a compression of your nerves that come out, then you get an itch in this spot right here. Is that true or not? Oh, there's the answer. Yeah, don't turn
it. Went off. There
Oh,
you go.
Excellent.
I was at the same talk.
Oh, cool.
Yeah. And he was talking about the spinal collapse at the two lower spinal vertebrae that would cause the...
And then does that have referred pain or
So he had
referred itch?
Yeah, exactly.
Is that what he's talking about? Yeah.
Then he had the other one where they had the spinal, high up in the spine where they would get the itch on the lower antebrachial areas and the lower legs as well. Yeah.
Probably.
But I think it's amazing that there's so many similarities.
Absolutely.
And that the skin is such a beautiful organ. And then there's quite a lot of discussion about melanoma, of course.
Yes.
And not every melanoma in animals is good. As a matter of fact, we have really malignant ones that really look like
Is that right? Okay.
the human melanomas, but they're very specifically located. So in dogs, for instance, when they're near the nails, when they're near the mucocutaneous junctions or in the mouth, they're very, very aggressive. Well, often in the skin itself, they're not. So
I see.
yes, please.
In dogs and cats, surface cytology is very important for diagnosing and treating disease. We will take little acetate tape and we'll stick it to the skin, and then we will stain it and look under the microscope and observe bacteria or yeast, and that will help us treat the animal. Now, my technicians, my staff members in the clinic will sometimes complain of an itchy rash or something on their skin. And so, of course, I do the same thing. I stick tape on my staff members and look under the microscope. And sometimes they will have malassezia on their skin. Is malassezia an important thing in human dermatology like it is in dogs and cats?
Yes. Our body, it's a kind of a commensal organism, so we have it. Usually, just like what we were talking about, our body or our skin knows not to react to it. But for some patients, yes, they induce an immune response. So seborrheic dermatitis, for example, so not everyone has the seborrheic dermatitis, but they do, or severe seborrheic dermatitis on their scalp. So it seems like for whatever reason, something tips some patients off, and then that induces inflammatory dermatitis.
Okay, one more question. I think this is the row of interrogators. Just speak to it.
Yeah. So earlier today we had a veterinary dermatologist talking about malassezia dermatitis and allergy testing. Is that something that's being done in humans where they're testing for malassezia hypersensitivity?
We don't really too much. In fact, for malassezia, we rarely even scrape or try to do that little tape testing, which we should. That's very easy. In general, for seborrheic dermatitis, we diagnose clinically. Rarely do we do any kind of scraping or biopsy or anything like that. In general, I'm not sure why.
All right, question in the back.
Kind of along that note, I'm sure this has happened to some of you guys when, I guess in your practices, where an owner will come to me and say, I have this rash on my arm or on my leg or wherever, and they'll show it to me and say, my dermatologist said that it's from my dog or from my cat. Can you tell me if they have this? And the dog or cat is completely normal physical exam. They'll tell me you need to scrape it, and can you take a biopsy, which typically will involve sedation and putting the animal at a bit of a risk to biopsy the dog's or cat's skin when there's absolutely nothing wrong with the dog. There's no clinical signs of scabies or whatever the dermatologist has suggested that the owner has. And so my answer typically to them before I will sedate their dog or cat is go back to your dermatologist and ask them to test you for that. So you just said that, clinically you diagnose things quite a bit clinically. When I guess are there tests for scape? Why wouldn't you not biopsy the person instead of asking us to biopsy a pet that is clinically normal? Not you specifically, but but this has happened 5 or 6, 7 times in my 18 years so far of practicing, and I have done it because the owner's like, I don't care, do it, and I'm like, well, okay, well, here are the risks, and yep, here's the negative biopsy. I have never gotten a positive biopsy or skin scraping or whatever I've done to satisfy the owner and the dermatologist.
Yeah, I think you're doing the right thing because probably the dermatologist, the human dermatologist, they don't understand what it's required to do a biopsy or scraping on an animal. I think we live in our own world, and that's why this is so important for us to interact, because the person who's sending you the pet and the owner to say, oh, just go, because they're thinking, oh, it takes me 5 minutes to scrape and do a biopsy on humans, so why not test it? They don't probably have a full understanding, because I'm sure if they did and knew what it took to knock the dog out or cat out, I mean, who would want that? So in general, and same thing in human dermatology, many times, yes, we can do the scraping for malassezia, but it's fairly easy clinically to diagnose and to try a certain medication. And I don't know if you do that in veterinary medicine, but oftentimes in dermatology, the hospital doctors laugh at us because they're like, you study that hard to do 2 things, either antifungal or steroid. It's like, so oftentimes we do just try and see if things improve. Unless we're really worried about something like melanoma or skin cancers. Oftentimes if someone comes in with dermatitis, whether it's eczema or allergic contact dermatitis, really the treatment for humans is topical steroid. So oftentimes we'll try it, and if it doesn't improve, then we may do a biopsy.
I think it would be scary to ask this audience how many people self-treated some skin lesion on their own.
Right. Yeah.
Probably everybody.
It's like me doing my dog, right? Putting that.
Exactly. And then also how many of these people have contacted some skin disease from either their animals during their career, because the fungal infections. Who has had a fungal infection from an animal in this audience? Has or had? Has or had? Okay, come up with those hands. All right, not now. Yeah, see, there's quite a lot of them. So, okay, another question there.
I'm curious, you mentioned acne is not an infectious disease. We see lots of pustules in puppies and other species. Can you just give a brief description of the cause of acne and how y'all treat it?
Yeah, we don't really understand the cause of acne in humans. There are 4 or 5 possible etiology or factors. We know that if you study acne, the cell proliferation, the epidermal proliferation is different. It doesn't occur normally, and that they tend to that's why you kind of clog up the pores. And then sometimes that's why retinoids work very well, because it kind of sloughs that first layer off. And it also, often retinoids are known to regulate differentiation of keratinocytes. So that's one possible cause. Second is P. acnes, but in humans, P. acnes is a commensal organism. So why we react to commensal organisms, just like Malassezia, for some people it's not clear. So is it truly an infection if you're responding to something that's on your body, like seborrheic dermatitis? We wouldn't call seborrheic dermatitis an infection. Third is probably somewhat related to oil, sebum. There are studies to say human sebum makeup is a little bit different than, I believe, some animal sebum. And there seems to be in acne sebum high levels of free fatty acid, cholesterol, and lower level of linoleic acid. And in the 70s, some people have thought that linoleic acid is the good lipid, and that they would treat people with it or give, and that improved some, but that kind of, that fad sort of stopped. So some kind of sebum abnormality, maybe perhaps also hormones, because it certainly acne occurs in humans during certain times in our lives. So those are the 4. And then recently, what we're finding is that immune response of each person is very different to this P. acnes, the commensal organism. So those are sort of the 5 factors that we think about when we're thinking about acne. The pustules are sterile in that they don't really harbor staph or anything like that, at least in humans.
All right. Once again, thank you so much.
Thank you.
And thanks for coming. Wonderful, wonderful, wonderful. So